Open the pod bay doors, Doc.
I'm sorry, Dave. I'm afraid I can't do that.
What would you do with a brain if you had one?
Do? Why, if I had a brain, I could...
Miles per hour.
I could while away.
46, 56 degrees.
Hello, and thank you all so much. I'm really excited to be here. In fact, only a few months ago, I was telling a good friend of mine, Chris Schmidt, who's up front here, that I really needed to watch more John Waters movies, and this was the perfect opportunity to watch Polyester. So, a lot of my research focuses on this question of whether or not there was a trade-off between vision and olfaction or smell in primate evolution. This is a long-standing hypothesis that, in fact, primates traded a sense of smell that is very, very strong in mammals for better vision. So some primates, actually many primates, do have a very specialized visual system. So all primates have forward-facing eyes. This is a shared trait that all primates have. Many primates have very high visual acuity, so they're able to see detail. And some primates can see in trichromatic color vision, which means that they can see in 3 color channels, blue, green, and red, which allows us to see color the way that we do, at least those of us who have full color vision. Now, some primates also have traits that indicate less reliance on smell. So some primates, like this mangabe, has a very short nose. The brain of some primates has a small olfactory bulb. These are the olfactory bulbs in a human brain. They're really tiny, you can barely see them. But these are the olfactory bulbs of a mouse brain. They are very apparent. We also see primates that have fewer functional genes for olfactory receptors. So this is just a little figure that shows a mouse compared to a macaque chimpanzee and a human divine. And the red in this pie chart indicates intact olfactory receptor genes. The green in this pie chart indicates intact genes for being able to detect pheromones. The blue and yellow represent genes that are pseudogenized or don't work. And we see that in primates, there is much more blue than red, and there's much more yellow than green, which means that there are fewer functioning genes for basic smell and also pheromone detection. Now, this is a family tree of primates showing all the major groups. So we have aye-ayes, lemurs, and lorises, and also tarsiers, New World monkeys, Old World monkeys, and the apes, which include humans. Now, these two groups are quite different. So, we have the wet-nosed strepsirrhine primates that are the lorises, lemurs, and aye-ayes, and the dry-nosed haplorhine primates, which are tarsiers, New World monkeys, Old World monkeys, and apes, including humans. Now, my approach to my research has been to look at comparative morphology in living species. I'm also really interested in how genetics underlies the morphology. So how do genes code for the sense of smell? And I'm also really interested in the evolution of smell. So I'm going back to the fossil record and looking for signs of any features that are related to smell in primates. And this is where paleontology comes in. And so I use these approaches to study olfactory system evolution. Now, the olfactory system is actually comprised of 2 systems. The main olfactory system, which I will call the MOS, and the vomeronasal system, which I will call the VNS. The main olfactory system is in the main olfactory epithelium. You also have the olfactory bulb, which communicates with the main olfactory epithelium, and this is an extension of the brain. The vomeronasal system includes a vomeronasal organ that sits at the base of the nasal cavity, and the main processing center is the accessory olfactory bulb, which is a projection on this olfactory bulb. Now, how does smell work in a very simple explanation? So let's say we cut away part of Divine's face, or Francine Fishpaw's face, so that we can see inside. So inside of her nasal cavity, we see main olfactory epithelium that lines the nasal wall. The olfactory bulb is a projection from the brain that emits nerves into the main olfactory epithelium. And there are specialized cells called olfactory receptors in the main olfactory epithelium, which are coded by olfactory receptor genes. This is a relatively passive system. So let's say there are odor molecules from these flowers. When you just breathe in and out, you are passively inhaling those odorants, and those odorants will bind to olfactory receptors in the main olfactory epithelium and send signals to the olfactory bulb. Now, there's another kind of olfactory system, the vomeronasal system, that seems to detect pheromones, and pheromones are odorants that code or that have social and sexual signals. So the vomeronasal system seems to be really important in mating behaviors in many mammals. The main organ of perception is the vomeronasal organ, which is a tube-like structure that sits at the base of the nasal cavity. And it has epithelium lining this tube that contain vomeronasal receptors that are coded by vomeronasal genes. Now, again, the signals that the vomeronasal system detects are pheromonal, and they are sexual in nature. So this is a female ring-tailed lemur making scent marks on a tree. This is maybe a male. He will go up to that scent mark, investigate it, and that will send signals to the accessory olfactory bulb. A lot of mammals, when they detect pheromones, make this face. It's called flamin' face. Primates don't really do this, but many mammals do this because it helps get that those odorants into that vomeronasal organ when they go like this. It seems like the two olfactory systems are differentially tuned, where the main olfactory system is tuned for conditions that, or responses that are conditioned over life. And these can be ecological cues, so what does your food smell like? These could be predator cues, so does a predator smell a certain way and should you run away from that smell? And the main olfactory system can probably discriminate sociosexual cues as well. The vomeronasal system, on the other hand, seems to be tuned to more innate responses that are fine-tuned, so these are not as broadly tuned as main olfactory signals. And the fine-tuning also seems to relate to odorants in the vomeronasal system being more of a sociosexual or predator cue nature. And this is a system that I focus on. So this is the vomeronasal organ, and here I am slicing through the middle so that we can see into the organ. So This is what the organ looks like if you were looking at it head on, and this is from a mouse lemur. So this is from the nose of a little mouse lemur. They're really tiny and cute. So these are the vomeronasal organs, and they happen to be soft tissue only. So these are epithelial tubes that are surrounded by cartilage, and then the cartilage articulates with the maxilla, which is bony. And it just so happens that if you look at a slide from histology here, and then look at CT scans, there is a groove in the maxilla that is formed by the cartilage surrounding the vomeronasal organ. So we have this bony correlate of a soft tissue organ that we can potentially find in fossils. Now, across primates, the vomeronasal organ has several character states. So in non-primate mammals, as well as lemurs and lorises, the strepsirrhines, the vomeronasal organ is very well developed. In tarsiers and New World monkeys, two haplorhine groups, the vomeronasal organ is comprised of only sensory epithelium, and in some New World monkeys, it is more degenerated. In apes, the vomeronasal organ is vestigial, so it is present, but it doesn't seem to function. And Old World monkeys seem to have lost the organ completely. So we see that this system varies across primates, where strepsirrhine primates have a really well-developed VNS, tarsiers and New World monkeys have derived less well-developed vomeronasal systems, and Old World monkeys and apes do not have a functional vomeronasal system. So how did this evolve? So are there bony correlates of the vomeronasal organ that can be found in fossils? Yes. I worked on this for my dissertation when I was in grad school. I got histology specimens from a colleague, and then I CT scanned a bunch of primates at the American Museum of Natural History, and I was able to reconstruct them using software so that I could visualize the vomeronasal groove and other traits. So the vomeronasal groove is this shiny cutaway object here, and the vomeronasal organ is shown in these histology slides in B, C, and D. So the opening of the vomeronasal organ happens around here. We can see a small lumen. The midpoint would be this point in an actual specimen, and the end point is here on an actual specimen. And the vomeronasal organ and the vomeronasal groove are tightly correlated, so there is a strong relationship between these two features. So I measured the length of this vomeronasal groove, and in a publication from 2014 with a colleague, we basically looked through the numbers of published vomeronasal receptor genes across mammals in the literature, and we decided, let's see if these two features are related. So we looked at size-adjusted vomeronasal organ length from the vomeronasal groove, as well as the proportion of intact V1R genes that were found in published studies. And there's a very strong positive relationship between these two features. So I can go ahead and say that I can look at the length of this feature and try and find this in fossils. So as part of my lab, I had a former undergraduate, Becca DeCamp, who graduated in 2020. She's now a second-year PhD student at Rutgers. She helped me measure the vomer nasal groove length in fossils, and we also found more V1R gene numbers from the literature. And Becca here created ancestral state reconstruction models for V1R gene evolution in primates. And we used fossil specimens like this that had a really well-preserved palate. And Becca found that when we compare primates to outgroups, so the bottom lines here are several outgroups. This is a flying lemur, a tree shrew, a rabbit, and mouse. We found that V1R gene number with the proxy of the vomer nasal groove length was very high in mice and rabbits and reduced slightly in the common ancestor of primates and our closest relatives, the tree shrews and flying lemurs. And so this is generally where we approximate V1R gene numbers to be in the last common ancestor of mice, rabbits, and primates. In the last common ancestor of primates, we see that the number of V1R genes decreased slightly. There were V1R gains in strepsirrhines, especially in mouse lemurs. And we see V1R losses in the dry-nosed primates, the haplorhines. So, my general hypothesis is that old world monkeys and apes may use visual signals more often in mating. So, we know that female old world monkeys and some apes have visual signals of fertility, including sexual swellings. So this is the sexual skin of a baboon that is very swollen. This basically signals that this female is ovulating, and this is a sexual swelling on a female bonobo. Again, this is a signal that she might be ovulating, and males will take interest and they will mate with females when they have those swellings. Males also have visual signals that may indicate dominance. So mandrills have very colorful faces and butts when they are dominant. We see a similar feature in vervet monkeys. So male dominant vervet monkeys have bright blue testicles. The more subordinate vervet monkeys have browner testicles. So males have these visual signs of dominance that they can see from pretty far away. So we do also see higher rates of intermale aggression in old world monkeys and apes when they compete over females. Old world monkeys and apes have high visual acuity, so they can see in great detail, and they also have trichromatic vision. So maybe visual signals are something that you can see at a distance and may reduce the need for physical aggression during mate competition. If you think about a male going up to a female and sniffing her, that's getting really close to that female, and that might invite aggression from other males. But when males can use visual signals that can be detected from further distances, that may confer a benefit that males don't die during competition over females. So I happen to think that maybe not all of the olfactory system was reduced in primate evolution, but we do have the vomeronasal system becoming less important as visual signals become more important in mating in these old world monkeys and apes. So I want to thank you all so much. Thank you, Beth and the Coolidge Corner Theater so much, and thank you to my lab and all my collaborators. and enjoy the movie.